Welcome to a walkthrough of WEGAN's ecological data uploading process. WEGAN is a web-based platform for analyzing ecology data that requires no coding knowledge. In this step-by-step tutorial, we will show users how to upload their own ecology data for analysis. Let's begin.
For this data uploading example, we will work in the correlation module, although the uploading process is similar for all modules. We first arrive to the Upload Your Data page. Here, users can either upload their own ecology data or analyze sample data sets. Community abundance data is essential to all modules. This data consists of sites or samples and species or taxa. Several different supplementary data types can be provided based on the analysis module. This diagram outlines the supplementary data requirements for different modules. Community abundance data is always required, and environmental data is helpful in all modules. Meanwhile, trait data can be optionally used in the diversity module, and taxonomic data can be used in several modules, but is required in the taxonomy module. To upload your own ecology data, click on the Choose File button. This should be the primary data describing community abundance, meaning sites or samples versus species or taxa data. The data should be in CSV or TXT format. After uploading your data, describe the format. Select whether the sample data is in the rows or the columns. The default configuration for community abundance data is to have species in columns and samples in rows, but ensure to select the configuration that matches your data. Next, select whether to retain the column and row labels. For example, say your format is species in columns and samples in rows. In your data, you are likely to have the species names at the top of each column, so you would want to retain column labels. If you have no site labels, i.e., the beginning of each row is the start of the data, and there is no header column designating the row labels in your data, then you would select only to retain column labels. For any dimension you do not retain the labels for, a pseudo-label will be used. To revisit these rules and recommendations for data uploading, hover over the question mark symbol. This opens a pop-up table outlining the recommended formatting. Click the Supplementary Data Tables banner to upload additional data, with upload steps similar to those for community abundance data. For correlation analysis, environmental supplementary data can be uploaded. Click Choose File to upload original environmental data. This should also be in CSV or TXT format. Select the format. The default configuration for environmental data is with variables in columns and community abundance data-matched samples in rows. Select whether to preserve row and column labels. When done with all data uploading, click Submit. If there is a mismatch between the community and supplementary data, a pop-up message will highlight the error. In this example, the number of rows or sites in the community data is not the same as in the environmental data. This was due to selecting samples in columns instead of samples in rows, when the overlapping site data was actually in the rows. The number of overlapping rows and columns must match before proceeding. This diagram describes the recommended configurations for community and supplementary data tables. When working in the taxonomy module, taxonomic data is mandatory in addition to community abundance data. You can still upload supplementary environmental data in the taxonomy module. In every module, there are also sample data sets available for use under the Try Our Test Data banner. Simply click a radio button to select a dataset. Selecting a dataset will also load its accompanying supplementary data. In this case, environmental data is provided. When working in the taxonomy module, taxonomic data will also be loaded automatically. If using your original data, click the first Submit button to proceed. If using a sample dataset, click the second Submit button to proceed. Next, we arrive at the Data Integrity Check page. This page tells us the results of our data pre-screening. If the data meets integrity criteria, users can proceed. If data does not pass the integrity check, users should adjust and reload their data. Remember to upload data in CSV or TXT formats and to organize overlapping data, such as site data, in the same format in both the community and supplementary tables. Missing values, if present, can still pass the integrity check and will be addressed on a subsequent page. Scrolling down, we can see a PCA plot of our data. We can also switch to the Heat Map tab for a heat map overview of our data. If necessary, data can be directly edited by switching to the Data Editor tab at the top of the page. Data editing options include changing the data type, editing the data, and removing data. Ensure that the data type for each variable is correctly categorized as either categorical, continuous, or ordered. For community abundance data, continuous variables should be selected. Any issues in the data types will raise an error message on the Data Overview tab. Click on the Edit icon to open a dialog box with sample data for a variable. Here, we can edit individual sample data for the selected variable, in this case, the agro.rupee species variable. We can also remove samples entirely by clicking the Remove icon. Any data removal will prompt a confirmation because it will remove the data for that sample across all variables, not just the variable being edited. Click Confirm to continue with the deletion or Cancel to go back. Once ready to continue, scroll down and click Proceed. On the Missing Value Estimation page are instructions on how to deal with missing values. Note that you can locate this page manually through the navigation tree on the left-hand side. We can first select whether to remove species that are missing more than a certain amount of data, and can set that threshold manually. Secondly, we can choose a method for filling in missing values. Options include replacing missing values with half of the minimum positive value in the original data, replacing missing values with the variable's mean, median, or minimum value, or estimating the missing values using KNN, K nearest neighbor, PPCA, probabilistic PCA, BPCA, Bayesian PCA, or SVD, singular value decomposition. After making your selections, click Submit to continue. We next arrive at the Filtering Rare Species page. Rare species can be defined based on their presence across all samples. Here, we can manually adjust the threshold at which we differentiate between rare species and noise. Users are encouraged to apply thresholds based on their analysis goals. Adjust the slider or manually input the abundance threshold at which to retain species. The default is 5%. Click Submit to continue. On the Normalization Overview page, users can perform normalization procedures on our community abundance data to make it more comparable. Select a data normalization method to adjust the data distribution. Select a data transformation method to reduce skew or meet model assumptions. Select a data scaling method to adjust the data to a specified range so that magnitudes are comparable. Select a hybrid method to apply normalization across more than one dimension, or to normalize across one dimension and also transform data. Click the Manual button to apply your selections.
Choose Automatic Normalization
to automatically obtain the optimal normalization based on Pearson mode skewness. A pop-up will show you variable data distribution plots comparing the effect of your normalization on variable data. Move to the Sample Overview tab to see similar plots for your samples. These are the data distributions after our manual normalization. To save your data distribution plots, click the Export button. In the Graphics Center, we can adjust the export configurations. Select the export format from a drop-down menu. We can select from PNG, TIFF, PDF, SVG, and PostScript formats. Next, select your desired export resolution. Options range from 72 dpi to 600 dpi. Finally, select an export size of either full page or half page. Click Submit to generate the export file. The download link will now update. Click it to open the export file in a new tab. After normalizing the data, click Proceed to continue. This second data integrity check page is for our supplementary data, in this case our environmental data. We see that our environmental data has passed the integrity screen. Click Proceed to continue. If other supplementary data is present, it will also have a separate data integrity check. This page is for the taxonomic data. Since our data passed the check, we can proceed to the next page. After completing the data pre-processing steps, you can now proceed with analysis-specific steps. Each module has unique analysis options from this point forward. You can continue to any of our module-specific walkthroughs to understand how to proceed with your desired analysis method. We hope you found this data uploading tutorial helpful.